Hi, welcome back to Biology. My name is Mr. Kubuske. Today we're diving into Unit 5. Unit 5 is all about DNA. Actually, our next two units are going to be about DNA. This one's more about discovery, structure, and how we make DNA. The next one will be like protein synthesis. We'll talk about how we take this DNA code and turn it into a protein that's usable by ourselves. Today we want to answer three big questions. The first one is what scientists contributed to DNA's discovery, what's the composition of DNA, and what is the shape of DNA. Okay, so think about those as we go. We're going to be focusing on the scientists, and as we go through, we're going to talk about the experiments that each scientist did in order to contribute to the discovery of DNA, and then as we go through, that'll help us answer some questions about DNA structure. So, the first scientist we're going to talk about is the one that's actually credited with discovering DNA, although he didn't know what he had discovered at the time. His name is Friedrich Miescher. Could be pronounced Miescher or Miescher, either way, okay? I'm sure he would have a preference, but I'm sorry. Sorry, Friedrich. Okay, anyways, he's a Swiss doctor, and he was obsessed with white blood cells, wanted to find out about how white blood cells worked and what controlled them and things like that. In order to get white blood cells, now remember he was a doctor, and we're talking about like Civil War times, like late 1860s. In order to collect white blood cells, he had to collect pus from bandages in order to study his white blood cells. And remember, like pus is that white milky material that comes out, you know, of a cut and things like that. It's white because it's filled with so many white blood cells. Anywho, what he did was he took those white blood cells, and I'm going to use my little yellow balloon here as a white blood cell, a leukocyte as it's called. And what he did, he put them into a hypotonic solution, which you remember from the last unit, causes cells to swell. And when they swell big enough, they burst. Now, the reason he did that was because he wanted to know what was inside, and I'm sure you probably couldn't see it go flying out in my movie here, but what came flying out of my cell was something Friedrich called nuclein. Nuclein was a material, that he really didn't know what the purpose of it was. He knew it came from the cells, he knew it came from their nuclei, but he didn't really know what it did or what it was for. So he just called it nuclein. And for almost a century, his work was, people understood that he had found something, but didn't think it was worth anything. They just thought it was like junk material inside the DNA. What they believed controlled your traits was protein, which, if you think about it from their terms, makes sense because you and I are made of protein. Everything about us is made of protein. So everything that controls us is protein. Well, but what makes the protein? How do we know what kind of proteins to make? What proteins determine that I have a reddish color hair and that you have blonde hair, black hair, whatever? Okay. So that's really what we wanted to get to. We needed to find out what actually controlled the traits. Was it protein or was it this nuclein that Friedrich had discovered? In the 1950s, a lot of things were happening with DNA, and that's what we had become to know nuclein as. Nuclein was changed to nucleic acid because of its composition, which we'll talk more about here in a second. And eventually we came to know it as DNA, or deoxyribonucleic acid. So anywho, Hershey and Chase in the 1950s did an experiment. A lot of people call it the blender experiment. And what they were trying to find, though, was they wanted to know what actually controls your traits. What makes you you? Is it protein or is it DNA? So what they did was they had viruses that they laced with radioactive isotopes. Okay, the first one they laced with a radioactive isotope that affected the protein around the virus. That would be our green around here. That's the radioactive protein. Okay. Now, what should happen if protein controls everything about an organism, then when a virus goes to infect a cell, okay, because cells are actually what are used to make more copies of the virus. Viruses don't make copies of themselves. Okay, when we make copies, okay, what should happen is that the new viruses, if protein is controlling it, should look exactly like their parent. All right, let me turn this around so you can see it. Now, if I turn around and look inside the cell, the viruses that were created don't have that green coating around them. So that tells us that this green protein was not passed on to its offspring and therefore is not the genetic material. Likewise, they did a second experiment, and this time they put radioisotopes into the DNA. They made the DNA radioactive. Okay? Same theory applies. When the virus infects this cell and it passes on its traits, then the new viruses, if DNA controls them, should also have radioactive DNA. And if I spin it around and look inside the cell, sure enough, oh, let me move the other way. Look at the offspring compared to the parent. They're exactly the same. So that told Hershey and Chase that this radioactive DNA, or DNA in general, is what is controlling our genetic material, or determining our traits. It's the DNA, not the protein. So this settled that debate once and for all. So thank you, Hershey and Chase. And no, they didn't make candy bars on the side. But this still leaves us with a big question. If DNA controls everything about us, well, what is it? Like, what does it look like? What does it do? How do we make it? Etc. Where can I find it? Okay. Well, there were lots of scientists, like I said, in the 1950s and 40s doing experiments like this on DNA to find out more about it. So here are some things that we found out during that time. First of all, like I said, we're going to call it deoxyribonucleic acid. Now, DNA is made up of nucleotides. Remember, all organic compounds are made up of some kind of building block. The building block of DNA is a nucleotide. Now, each nucleotide is composed of 3 parts. A 5-carbon sugar, known as deoxyribose. Remember, if it ends in ose, that means it's a sugar. That's from Unit 2. A phosphate, and then 1 of 4 nitrogen bases. So it could be sugar phosphate and adenine, sugar phosphate and guanine, sugar phosphate cytosine, sugar phosphate thymine. But 1 of the 4 was added to it. So that group of 3 is known as a nucleotide. That's very important. You may want to put a star next to that or something. Okay? But it's made up of sugar, phosphate, and base, SPB. Now, why 4 bases? What do those have to do with anything? Well, scientists just kind of assumed that they just like went in a pattern, like it went A, G, C, T, A, G, C, T, A, G, C, T. But that would mean that everybody's DNA is exactly the same. And if everybody's DNA is the same and that controls our traits, then that means everybody must be exactly the same. But we obviously know that that's not the case. So leave it to this scientist by the name of Chargaff, and what he did was he measured the amounts of the 4 different types of bases in different types of organisms. In our class, we're going to do a you try it, okay? What we'd have are 4 different colors of paper here. If you count them out, okay, and add them to a table, okay, you can find some relationships between the bases. And the relationship that Chargaff found was that for the amount of adenine, however many adenines there are, there's almost always the exact same amount of thymine. Now I say almost always because in real life there are mutations that occur which change and pair things up the way they shouldn't be paired. But the rule became that A is always paired with T, adenine is paired with thymine, and G is always paired with C, or guanine is always paired with cytosine. Okay? That's the first thing we can determine from the bases. Okay? So those get paired together. The second thing that we were able to figure out from this is that G and A have similar shapes. They're larger than T and C. So we call them purines. Purines have double rings, and an easy way to remember which 2 of the 4 are purines is the phrase grapes are pure. G and A are purines. Likewise, on the other side, there's pyrimidines, which only have the 1 carbon ring. Okay, and an easy way to remember them is crazy tall pyramids. C and T are pyrimidines. Okay, now a purine always bonds with a pyrimidine. And what we'll find out is that A has to bond with T because of the number of bonds that occur in between them. So A can't bond with C even though one's a purine, and one's a pyrimidine. So this proves to other scientists, and is going to help us unlock the shape eventually, this proves that there have to be 2 complementary sides of a DNA molecule. If I have T on this side, then on the opposite side has to be an A. They are paired together. That would be the only way that they would have equal amounts in nature. Same thing with C and G. If I have a G on one side, then the opposite side has to say C, and that's how they get paired together. But we still don't know what DNA looks like. So let's get a picture of it, right? Let's take a selfie. Rosalind Franklin was hired by King's College in England to work with a guy by the name of Maurice Wilkins. And what she did was she took X-ray diffraction photos of DNA. Now, if you're using your iPad to take these notes, I would suggest going to the photo booth app and actually taking an X-ray photo of this, what's called photo 51, this picture right here. Because this is what actually unlocked the key to DNA. But unfortunately, Rosalind didn't get a Nobel Prize like some of these other people did for her discovery. The problem was that she had actually passed away before they could award the prize, and they don't award Nobel Prizes posthumously or posthumously. And that means that she was already dead before they had passed out the award. And the worst part was that she had contracted cancer. That's what she died from. And chances are she probably got the cancer from being exposed to these X-rays. So her work actually was what ended up killing her. And to top it off, the guy that she was working with, this Wilkins guy, it's widely believed that he actually stole her work. Now we're going to say it, and we're going to hope that that was not the case, and we're going to say that she gave him one of the 2 pictures that she took. But like I said, it's widely believed that he probably stole it from her and tried to take the credit all for himself. So anywho, so she took 2 pictures. She kept one of the pictures and tried to solve the mystery herself. She kept a blurrier one, thinking that it had more information. She gave a clearer picture, what we come to know as Photo 51, to Wilkins, thinking it was worthless because it only had a simple X and a big circle. That can't mean anything, right? But this circle and this X actually ends up showing us what the shape of DNA is. So Wilkins takes this picture and he goes to these other 2 colleagues of his, James Watson and Francis Crick, who are also working on the shape of the molecule of DNA. And we credit them with being the scientists who correctly identified the shape of the DNA molecule. Now, how they did it was they took the pieces of all these different experiments as like pieces of a puzzle and put them all together. They used Chargaff's rules, A to T and G to C, to tell them that it had to have 2 strands. They used the X in that X-ray that Franklin took, and that tells them that it's a helix shape, meaning that it's like a spiral spinning away from you. Okay? And they also took the information that it was composed of a sugar, phosphate, and a base, SPB. Okay? So I want you to take a second, maybe pause this, and think about using those pieces of information, either A, or what the shape might be if you don't know already, or 2, see if you can figure out like how, knowing what the shape actually is, why it makes sense that they came up with the conclusions that they came up with. The shape that they came up with was known as a double helix. Okay, that's the reason, if I go back to this, it's the reason that it's a perfect circle. There's actually a helix starting on this side. At the same time, at the same height, there's a helix going the opposite direction. That's why it looks like it fades and then it gets brighter again. Go back. So the double helix means that there's 2 helixes like spinning around one another. In the center, like rungs of a ladder, are the bases paired together. A on one side, T on the other, G on one side, C on the other. Okay. They determined that A and T can have 2 bonds between them and no more, 2 hydrogen bonds, and that G and C have to have 3 hydrogen bonds between them. So that means if I need 2 bonds but something else has 3, that they can't get paired together, which is why A has to bond with T and G has to bond with C. They determined that for each one of these bases that they're bonded to a sugar, which is then bonded to a phosphate, and then a sugar, and then a phosphate, and a sugar, and a phosphate, and a sugar, and a phosphate, and they make up the rungs of the ladder, the sides of the ladder, or the actual helix is made of the sugar and the phosphates. They also figured out, which is pretty cool, that the 2 helixes are anti-parallel, meaning if one's going up, the other one is pointing down, and the information goes in that direction. So if this were an accurate picture right here, the A and the G would actually have to be upside down, and the T and C would be right ways up. And they called their model intelligent because the pattern was random, but that pattern was like a code telling us what kind of protein to make, and that actually ended up being the code for all of our traits. So the important thing about this, and the reason that it was so widely accepted, is because A, it followed Chargaff's rules, A to T, G to C. B, it matched Franklin's photo. That X shape told him that it was a helix. And 3, it means it's intelligent. It's passing on genetic information, which is what Hershey and Chase said. So the final version, or final simple drawing of a DNA molecule can be found on page 591 if you're using the Pearson iBook. And I would suggest, if I'm doing these on Notability, to copy that picture and paste it into my notes so I have it. And then if it's not labeled, please make sure you label it. Sugars, phosphates, and bases. And then make sure you label the bases, adenine, guanine, cytosine, thymine, and then put whether they have 2 hydrogen bonds or 3 hydrogen bonds. So, to conclude, to wrap up, what scientists contributed to the discovery of DNA? Okay, there was Miescher, there was Chargaff, there was Hershey-Chase, Rosalind Franklin, in some ways Maurice Wilkins, and then Watson and Crick. Okay, what is the composition of DNA? Sugar, phosphate, and base. It's made up of nucleotides. Sugar, phosphate, base. Sugar, phosphate, base. And then what's the shape of DNA? It's a double helix. It's a twisted ladder with the rungs in the center being the bases, and then the ladder, the sides of the ladder, being the sugars and the phosphates. Sorry if I talk too long. This stuff's pretty interesting. It's so exciting. It's all this like treachery and mystery, and it's just a cool topic to talk about just because of it's amazing to think that you and I, the intelligent creatures that we are here because of this little simple molecule, and just happen to be the combination of these 4 little bases tells us that we become what we become. So anyway, I look forward to seeing you on the next one. On the next one, we'll talk about how to make more DNA, and we'll see you then. Have a great day.